we just talked about the chloroplast and the mitochondria, we're going to take a second to look at what we mean by this endosymbiotic theory. The endosymbiotic theory attempts to explain how we ended up with organelles like the chloroplast and the mitochondria inside of cells. If we break this word down, endo means inside, and symbiotic means that you have two things that are living together. So let's take a look at what this says. It says the mitochondria and the chloroplast were once primitive single-cell prokaryotic cells, but they got engulfed by a larger prokaryotic cell and developed a symbiotic relationship. According to this diagram, you can see that you have a larger cell, and it's initially lacking mitochondria and chloroplast. And you can see that it's engulfing this aerobic prokaryotic cell, which means it has the ability to make ATP, and engulfing this smaller photosynthetic prokaryotic cell, which means it has the ability to make sugars through photosynthesis. And then we just created a new place for these smaller cells to live inside of a bigger cell. As for everything in science, our theories have to be supported by a substantial amount of evidence. So here's the reason why we believe that the ancestors to mitochondria and chloroplasts were once primitive cells that lived on their own. One, if you take a look at bacteria, a prokaryotic cell, we know that it's going to have circular pieces of DNA inside of it, and we find those as well in mitochondria and in chloroplast. So they both contain circular DNA, and DNA codes for proteins. Those proteins are made by ribosomes. We talked about prokaryotic bacteria cells have ribosomes to make their own proteins, and we also see that the mitochondria and the chloroplast contain their own ribosomes, and they can produce their own proteins. We know they're about the same size and shape. We can analyze their DNA, and we find that they have common genes. Common genes indicates a common ancestor. So our modern day bacteria and mitochondria that we find in eukaryotic cells, they had a common ancestor. Also, their ribosomes are about the same size. Prokaryotic ribosomes are a tad bit smaller than eukaryotic ribosomes are. We're going to see in a future unit that when bacteria cells go to divide, they elongate, they copy their DNA, and then they pinch into two individual cells. We see that same type of division in the mitochondria and the chloroplast. When bacteria cells divide, it's called binary fission. Bi means two, so we have one cell splitting into two, or one mitochondria splitting into two. Fission means to separate. And then the final piece of evidence that we're going to discuss is that if you have a smaller cell, it's going to have its own membrane, its own plasma membrane. And this larger cell also has a membrane. Whenever a large cell engulfs a large particle, then it ends up wrapping its membrane around it and bringing the structure inside. And now this structure has two membranes on its outside. And when we take a look at both mitochondria and chloroplasts, you can see that there's two membranes. Here is showing you the inner and the outer membrane in a chloroplast, and then our notes we talked about the inner and outer membrane of that mitochondria. So they have two membranes because they were engulfed by this process called endocytosis. And you don't have to remember endocytosis, it's something we're going to talk about next class. But we're going to have two membranes around those structures. Now we're going to take a look at cell parts that are involved in transport. When I'm talking about transport, I'm going to talk about transporting things in the cell, and we're also going to take a look at how individual cells are able to be transported from one part of the body to another part of the body using these cellular structures we're going to discuss. So the first cell part we're going to talk about is the cytoskeleton. The cytoskeleton serves as highways that vesicles are moved. So you learned that the rough yard modifies proteins and ships it to the Golgi. Well, how do these vesicles that contain proteins and lipids from the smoothie are, how do they get to the Golgi? How does the Golgi send it to the plasma membrane? Well, these vesicles are walked along these tracks that are called the cytoskeleton. So that's where we say they serve as highways at which molecules can be moved, and even organelles can be moved and walked along the cytoskeleton. They also provide structure to the cell. Think of these cytoskeleton as like beams that are holding the ceiling up in a room. So it provides some structure to the cell. You're less likely to apply pressure and be able to break the cell because of the cytoskeleton. It's also used during cell division. And we can see that happening here. Here a cell is about ready to divide. And we can see that we're moving things, those are chromosomes, we're moving things around in the cell so we can split the cell in two. And we have the assistance of cytoskeleton. So these beams right here are part of the cytoskeleton, and we're using them to move different parts of the cell around so we can split the cell in two. When we look at cytoskeleton, we're usually looking for beams that are crisscrossing all over the cell. Now, another cell part that assists in cell division, or the movement of molecules around in the cell, are the centrioles. So the centrioles also assist in cell division. We're going to talk a lot more about these structures, but for now, all you need to know is that they occur in pairs. They look like short cylinders. Those are the cytoskeleton right there. Ultimately, you can see that they move to opposite sides of the cell, and they're going to assist in cell division. Next, we're going to take a look at structures that can help cells move or help move substance around in your body. In this situation, we're trying to move mucus. We're trying to move mucus up from your lungs to your throat. And we're going to use a cell part on these specialized cells that are called cilia. Sometimes you're going to see cells diagrammed like this with cilia on their surface. And this would be a simple unicellular organism that is using the cilia to move in one direction. So here we have a cell part that's helping an entire cell move. So this would be like a protist swimming around in pond water. It helps single cells move, and it also helps them acquire food. Now our spacing is off on the note, so I'm going to move down here in just a second, and I'll show you how the cilia would help a single cell protist acquire food. But let's take a look at this diagram right here. Cilia can help multicellular organisms move substances like mucus or eggs down the fallopian tube in females. What's going to happen is as you're breathing, air is coming into your lungs, into your airway, and you're breathing in dirt as well and other particles that you want to get out of your lungs. What the cilia does, and we have cilia right here, so let's go ahead and label the cilia. And notice it's only on one side of the cell. The cilia move back and forth. And what's that going to do, or what that will do, is it'll make the mucus move, and it's going to bring those particles that you just breathed upward into your throat. Cilia also aid in the movement of the egg down the fallopian tube. The fallopian tube has to get from the ovaries to the uterus, and the cilia will move back and forth, and it makes the egg move from one direction to another direction. Whenever we diagram cilia, they're usually short and hair-like you see down here. And notice not all cells have cilia that completely wrap around the entire cell. It just depends on the type of cell, whether it's going to have cilia or not. So moving down here, we can see I have a protist and it's covered in cilia. This is its mouth. And if there's food particles nearby, the cilia that line the mouth can flutter or move, and it can cause those food particles to move into that area that's considered their mouth. Now we're going to look at another cell structure that helps single cells move. And this structure kind of looks like a tail. It's called a flagella. And sometimes we see multiple flagella. Sometimes there's flagella in front of the cell, or what you would think would be like the head of the cell versus the end of the cell. And these things are going to move like the cilia and move this cell from point A to point B. They're much longer than cilia, though. Now I want to switch gears excuse me, and go into what's called the endomembrane system. The endomembrane system consists of these cell structures that can exchange materials through vesicle transport. You already have some background with this. We've already said that the rough ER can send modified proteins to the Golgi. The smooth ER can send lipids to the Golgi using vesicles. We know that the Golgi can send out vesicles to the plasma membrane, like you see here. We see that sometimes these vesicles will form lysosomes. So all these cell parts that are interacting through vesicle transport are part of the endomembrane system. So let's indicate these organelles. We have the nucleus, or the nuclear envelope, or the nuclear membrane. If you notice here, it just connects directly to the rough ER and the smooth ER. So they're all part of the endomembrane system. So we have the smooth ER, the rough ER, we have the Golgi, the lysosomes, the plasma membrane, because it can bring in particles using vesicles, or vesicles can fuse with the membrane, dumping contents out. And basically everything else that we have not listed here is not part of the endomembrane system. The reason why these can participate and exchange materials using vesicles is because they're all made of the exact same material. They are a single phospholipid bilayer. So they're all made of phospholipids on their exterior. So since they're made of the same thing, they can exchange pieces of one another. Again, my spacing was messed up on the notes, and I apologize. But I just wanted to show you that if I zoom here to this membrane, so I'm looking at a membrane right there, which is the outer part of this rough ER or smooth ER, then if I zoom in, then the structure there, this line is really going to look like this. It's going to be a phospholipid bilayer. It's going to look just like the cell membrane does. And because they're made of phospholipids in a bilayer, like you see here, and this is showing you what a vesicle looks like. You can see you have a phospholipid bilayer here. So this would have materials inside of it, like proteins that are being sent from the rough ER to the Golgi. They can exchange pieces. The structures that are not part of the endomembrane system would be things like the ribosomes. Ribosomes don't even have a membrane wrapped around them. They are not a membrane-bound organelle. The chloroplast and the mitochondria, they do have a membrane around them, but they have a double membrane, which doesn't fit with these other structures that just have a single membrane. So they are not a part of the endomembrane system. I've never shown you a diagram where a vesicle from the chloroplast is breaking off and like moving to the mitochondria. We've never seen anything like that. They're not part of the endomembrane system. The cytoskeleton would not be a part of it. Structures like the cilia and flagella would not be a part of it as well.